Welcome back to the continuation session. So, what we saw in my previous session, uh, we could identify a gene from cDNA library prepared from panicles of rice, and we sequenced it and did some bioinformatic analysis and derived a whole lot of information about its location in the rice genome, how many copies are there, and what kind of protein it codes by in silico translation, and then what that protein has in terms of functional domains. So what we found out that it has two domain like A20 and AN1 zinc finger domains, and then we studied the expression profile of this gene and find out that it is differentially expresses itself more in shoot as compared to root, and also the transcripts get accumulated more as compared to control under all stress conditions like salinity, drought, abscisic acid, and salicylic acid. And a kinetic study when we did, it revealed that it is its synthesis starts very early, within 30 minutes of the application of stresses, indicating thereby that this gene product is involved in very early events of stress management, like signal perception or signal transduction. And therefore, the gene became a candidate for further validation. Now, what we do in validation, either you overexpress a gene in transgenic scenario or you silence a gene. If you silence a gene, you lose the phenotype. And if you overexpress a gene, then you strengthen the trait to which it is linked. So let me share the slides and then explain to you how we went on. So this is the last slide I showed you in conclusion, that the expression profile of the gene indicated that it is involved in very early events of stress management, either in the signal perception or the signal transduction. Now, to validate what was done, the gene was cloned into a vector system. It has bucketing backbone, and you have the promoter and the terminator, and in between we put the coding region of the OSI ZAP8 gene in sense orientation as well as in anti-sense orientation. Sense orientation will overproduce, and anti-sense orientation will suppress the expression of the gene product. Okay, so this cloning was then confirmed by usual restriction analysis, and then transgenic plants were generated using particle bombardment system of gene delivery, and and the transgenic lines were generated. Now, if we did check whether the putative antibiotic resistant plants are transgenic and have the transgene integration. What we did here, that this study was done both in rice and in tobacco. It means that it is the rice gene which has been put into rice and rice gene put in tobacco. So both homologous and heterologous systems were checked. What happens when you take the gene from one system to another system. This also, if it works like a rice gene working in tobacco, it means this technology can be taken to any plant. So therefore, we checked or we validated this gene in both homologous, like rice gene into rice, and heterologous system, like rice gene in tobacco. Okay, so as a part of checking that whether the transgene has integrated and it is expressing and it is doing its job, what it is supposed to do. So in this panel, what you see is the tobacco transgenic lines 1 to 5 and the rice transgenic lines 1 to 5. So we picked up 5 plants, in fact, and then extracted RNA and then the rannitonic gel and transported onto the membrane. And so this is the tobacco, and the probe is the cDNA from rice. So when you hybridize, you can see all the transgenic lines have transcripts which are hybridizing to the probe, that is the rice gene. But in the control plant, there is no hybridization. It means since it doesn't have rice gene, so the control plant doesn't give any hybridization, indicating that these lines have integrated the transgene and it is transcribing, which you can see by this presence of this hybridization. Now, and these plots are done under very stringent conditions, wherein even if there is little bit homology, it is removed because of the stringent conditions. So only the rice gene will hybridize to its own complementary part here. You know, now when you look at the rice panel here, you see the control plants also are lighting up, but the transgenic lines are giving more strong signals as compared to the control. Now, this is because rice has its own gene. So it will hybridize to its own gene in the genome. And I mean, the transcripts made by the endogenous copy. And you can see the hybridization to in the transgenic lines is a lot stronger as compared to control, indicating that these plants have the additional copy in them. Okay, now one of the criteria which is used for uh, uh, stress tolerance is the chlorophyll uh, bleaching. Okay, so for example, these are the leaf bits from tobacco plants grown under normal conditions, and the leaf bits were exposed to different salt concentration from 0 to 200, 400, 600, and 800, you know. And you can see this is the leaf bits from control. And as the salt concentration increases, the cells lose chlorophyll. You can see visually, and at 800 millimole, the cell loses chlorophyll completely. Okay, so it shows that a normal plant, normal plant loses chlorophyll because of this stress given by the salt, high salt concentrations. But if you look at the leaf taken from transgenic lines, you can see these leaf bits are able to retain chlorophyll even at 800 millimole. And similar observations were made here when you do the similar experiment with rice, for example. These are the transgenic leaves from a transgenic line, and it retains chlorophyll, you know, even at the highest concentration, which was tested, 800 millimole, whereas the leaf bits from control plant, untransformed control plant, it loses the chlorophyll completely at 800 millimole. So what does this analysis means is that the transgenic lines which were generated are expressing, making transcripts, and also conferring tolerance against stress conditions of salt, you know. So, so this was an indication. Further, uh, when these plants were taken to greenhouse, in rice particularly, and they were given water stress, drought and salinity, what was seen here that the control plant dies within 20 days of the stress, and the transgenic lines, they survive and show the seed set. So seeds were collected, and then further analysis was done with the seeds obtained from the tissue culture regenerated plants, or what we call T0 generation. And these T1 generation seeds were assessed for salt tolerance, drought tolerance, and the cold tolerance. So if you look at this, as I mentioned in my previous presentations also, that one of the parameter which is looked at the seed germination itself. It is very, very sensitive for stresses. So if you see here, this is one in for rice and this for tobacco, and these are the control seeds germinated under stress, and they show almost 19 to 95% inhibition in seed germination, whereas the seeds of transgenic lines, they they those start slightly slowly, but after 7 days, they almost 100% seed germination was seen, both in rice as well as in tobacco. And if you see visually what is happening, this is the tobacco seeds after 7 days. This is control under stress condition, and these are the transgenic plants under stress conditions. They are as healthy as the normal plant, and this is the rice seedlings after 7 days old. This is control under stress, and these are the transgenic lines under stress conditions. They grow well, and also interestingly, they also show a better recovery. Means if you grow the seeds for 7 days and then stresses for 7 days and then bring it back to normal conditions and see how well they recover. So this panel in fact shows that. So this is the control plant. After stress, the plant was put to the normal conditions and it recovered after 7 days this much, and these are the transgenic lines recovering so well. So indicating thereby that this gene not only helps in salt stress tolerance, but also it helps plant to recover after the stress is over. Now, in plants, since they are sessile and they grow in one place, there is a mechanism which starts functioning to give them tolerance. And once these stress conditions are over, the machine gets back to the normal conditions. So that slowdown of the growth, which takes place under stress, has to recover back to the normal. So this panel indicates that this gene also helps in overcoming the stress conditions and recovery from the stress subsequently. Similarly, the temperature and the temperature stress. So similar results were obtained here on germination and then the recovery and all. I'm not going to spend much time on this panel, but we had the similar observations done for the other stresses like drought and the cold temperature, So, but then what was seen is the productivity part, when the plants were taken to greenhouse, and in greenhouse, they were exposed to salt and drought conditions, So these are the normal plants which were grown under normal conditions, and the transgenic plants grown under normal conditions. And these are the parameters they were assessed for, and if you see the transgenic plants, they match one-to-one with the controls grown under normal conditions, you know. But if you see here, they show a yield penalty of 52%. So even under normal conditions, transgenic lines are showing a yield penalty of 52% in the seed production. If you see the number of seeds per plant here and the number of seeds per plant here. Now, this penalty may be due to the constitutive expression of the gene, because that may be giving some stress of its overproduction. You know. But then, like if you come here, when the control plant exposed to stress of salt or drought, they die within 20 days. And you can see there is no growth. So there is no plant to compare with. And these are the different transgenic lines. And if you see here, transgenic lines under stress maintain the same level of yield as compared to the transgenic line growing under normal condition. Right? Shows only 50% penalty, which was observed here under both these stress conditions, indicating thereby that these overexpression of this gene is protecting the plants against both the stress like salinity and drought. You know, so to conclude this part here, that it means we have a gene in hand. If you overexpress it, it confers tolerance against abiotic stress of salt and drought and temperature. So, but then there are two more questions which we address to make sure that the technology is foolproof. In the sense, you know, this gene protein has zinc finger domains. And in general, if you look at the zinc finger proteins, they are DNA binding proteins and mainly referred as the transcription factors by and large. But now we know that zinc finger proteins have different functions, like in addition to DNA binding protein transcription factors, they are also RNA binding proteins, and also they take part in protein-protein interactions. So they have multiple roles. The zinc finger proteins have multiple roles other than DNA binding proteins or transcription factors. They are also involved in RNA binding and protein-protein interactions. Now, when we looked at the gene sequence of this, there is no nuclear localization signal, and there is no DNA binding domains. So these two observations told us that probably it is the cytosolic protein, but then it was contradicting this concept of zinc finger proteins being transcription factors and nuclear proteins, since it was not having any of these signals in the protein, no nuclear localization signal, and then no DNA binding protein. We concluded that it's a cytosolic protein. But then, so that is one part, that it indicated that it is a cytosolic protein. But then, what is this domain interaction? How the domain interaction is happening? See, only one protein, which was first reported, that is human protein, named as ZNF216. Now, this is a human protein of, which helps preventing N-kappa-mediated apoptosis. And this protein has both these AN1 and A20 domains, like what we have in our gene. And in this study, they used co-immunity precipitation assays, and they concluded that the A20 domain interacts with A20 and AN1. And it also has a protein kinase. So what is happening? This in vivo, it is a dimer where A20 interacts, and then A20 interacts with AN1, and I mean, it gets forced to itself, you know. So that is the kind of inference given for this human protein ZNF216 based on co-amino precipitation assays. So what we try to check whether this interaction is true by more direct evidences like yeast to hybrid system. And what is done in yeast to hybrid system, you know, is this. We amplified these regions, you know, AN1, A20, and AN1 regions. They were amplified and they were cloned in detailed vector and finally cloned into these yeast vector systems, and they were then co-transformed into EGY48 cells. And then they were selected for these interactions like beta-galactosidase or leucine prototrophy assays. You know. So how it is done actually, this is clear in this. So you have, you know, the yeast to hybrid system uses bait and prey concept. So this is bait and this is the target protein. So those domains were cloned here and here, which were amplified, you know, and the concept is if these two are interacting, then they will drive act on this promoter and drive the lexi gene or leucine gene expression. And if there is no interaction, then this will not happen. You know? So that's how it is decided. So if you look at those, I will explain you this panel. So what is done when you take co-transform cells, A20 and E20, means the bait and the target protein is the same, A20, A20, panel 1, and then A20 and AN1, panel 2, and then the reverse, you know, if you are taking a target protein as AN1, next you can take a bait as AN1 and target as A20. So this is panel 3, and then there are positive controls and the negative controls. So if you see here, you know, A20 when taken as bait and the target protein, they drive the expression of beta-galactosidase, and you can see this blue color here. Similarly, A20 and AN1, panel 2, they interact and drive the expression of the beta-galactosidase, and then AN1 and A20 means when you reverse it. And then there is no interaction when you take AN1 and AN1, and the positive control and the negative control. Okay, so this experimentation clearly demonstrated that these two domains, like A20 interacts with itself and also interacts with AN1, you know. So that story was clearly demonstrated. Now, another question which the conclusion which we draw based on the bioinformatic analysis, that absence of nuclear localization signals and the DNA binding domains indicate that it's a cytosolic protein. So the question was asked that how you demonstrate experimentally. So that was done using GFP. So GFP and SAP8 protein, fusion protein was made in the cell of Nicotiana lanthiana leaves. And this is the normal GFP protein, which shows its localization in the nucleus as well as in the membrane level. When you overlap this and this, you see things happen. But when you look at the hybrid protein, means the GFP fused with SAP8, you know, there is no nuclear localization here. All the fusion protein is localized or targeted to the membrane. That's all. You know, there is no nuclear localization. So this experiment clearly demonstrated that the SAP8 protein is localized onto the membrane, and it also supports its involvement in the very early event of signal perception, as I told you in the very second lecture, that signal perception is done at the membrane level, and then it is transduced to the nucleus. So this gene product, SAP8 gene product, is involved in very early events of stress management, which was indicated by the it was indicated by the Nathan analysis of the expression profile of the gene, that within 30 minutes it gets overexpressed in the cells. So indicating its role in the very early events of signal perception and transduction. And now its localization at the cell membrane level also confirms to the idea that yes, the gene product may be involved in signal perception or the signal transduction. So to conclude, what we have found here, that SAP8 protein is a single copy gene located on chromosome 6, which was isolated, and it was shown to be a multiple stress inducible gene, and it shows that it is involved in the signal transduction of stress signal, probably by protein-protein interactions, and overexpression of this gene in rice and tobacco confer tolerance to salinity and drought and cold stresses. So it means this technology can be used for conferring tolerance to other plants. So after the discovery of this technology, this has been used and tested and validated in other plant species, actually. And this is the most cited paper from us. And you can go through the details of this. I have put it on the Google class, this paper. For details, you can go through this paper. And if you have any questions, you can get back to me. And we will discuss it when we meet in the common meeting room.